I would like to just briefly say that there have been a number of drugs that have been developed at Scripps. I don't have time to go into them all, but I do want to highlight this one listed at the top. In March, the drug ozanimod was approved by the FDA for treating multiple sclerosis. And this has been long in the works, and we're very proud that this medicine was developed by laboratories at Scripps Research. We've also lately been organizing a lot of our efforts around infectious disease into what we call the Global Health Initiative. And this is in part led by Christian Anderson, but it's composed of a number of faculty at Scripps Research, at Caliber, which is the drug discovery engine founded by our president, Pete Schultz, and by the Scripps Research Translational Institute that was founded by Dr. Eric Topol. These three components of Scripps all are working together to put together a very comprehensive approach to infectious disease. And there's basically three areas that we can cover in mounting a coronavirus program. There's surveillance and diagnostics, there's development of vaccines, and there's development of antiviral therapeutics. Now, diagnostics they're actually, from a research standpoint, well in hand. We know how to detect viral infection using PCR diagnostics, and we know how to detect in serum if the body has mounted a response to the virus. The key issues with diagnostic are not really research, rather, but they are scaling up distribution to testing hundreds of millions of people. So we're not going to focus much on that, but rather we'll talk a little bit about these other three areas. So in the surveillance area, many of you heard Christian Anderson talk about how he uses genomic sequencing to build a family tree of the virus and actually track its spread across the globe and throughout the country. And he also did some seminal work on understanding the origins of the coronavirus. Eric Topol, who's director of SRTI, has actually used digital medicine approaches to look at viral infection. So they had a very successful study where you could tell people were about to come down with the flu by examining the fitness data on their personal fitness devices. And they're using this technology that they had developed for that application to look at coronavirus as well. Now, on the vaccine front, we really have what I would call the dream team of vaccine deployment. There are a couple people I'd like to single out. So Dennis Burton is working on developing vaccines, and he's also screening patients who have recovered from coronavirus for really potent antibodies in their bloodstream that can be used for neutralization of the virus. Andrew Ward, working closely with Ian Wilson here on the California campus, they are using both electron microscopy and X-ray crystallography to look at the complexes between antibodies and the viral antigens. And then we also have Hiren Cho and Mike Farzan, who have been collaborating for a number of years on coronavirus. And I'm going to defer further comments because I'm sure Mike will fill us in on their seminal vaccine efforts. Now, on the treatment, we also have a really important set of drug discovery programs. One is led by Arnab Chatterjee. He's the head medicinal chemist at the Caliber Institute here in La Jolla. They are using what we call the Reframe Library. This is a library of about 14,000 compounds who have been in either clinical trials or they're approved drugs. We can rapidly screen those drugs for antiviral medicines. There's a powerful screening effort going on in the Florida campus, led by Lou Scampavia and Tim Spicer. So they have a molecular screening center, and they're using high throughput campaigns to screen for antiviral compounds. And Matt Disney is using RNA drug discovery to find novel compounds that would target the RNA of the coronavirus. So there's a lot of activities in all of these areas. And I would really like to focus on Mike at this point. And Mike has joined the faculty. We were able to recruit him from Harvard. Mike worked extensively on the SARS virus. So SARS came around over 10 years ago, and it's actually quite related to the current coronavirus, COVID-19. And of late, Mike has been working a lot on HIV vaccines. And so these two things, so he was actually the person who discovered the ACE. It was the target of the virus trying to enter human cells. And his vaccine work and this prior work on SARS has positioned him extremely well to combine these two approaches, and so that he can actually mount a serious and rapid assault on the coronavirus using vaccines. And so I'm going to stop here without further introduction, and I'm going to let Mike take over and deliver his seminar.
Thank you, Jamie. Today I'm going to talk about how scientists and then scientists at Scripps are doing their best to address what we call the coronavirus challenge. And I do want to get a little bit of housekeeping out of the way first of all. So the first thing is, what are we going to call this thing? Because everybody calls it something a little bit different. The coronavirus, the virus, the COVID-19 virus, the SARS coronavirus, the SARS coronavirus 2, or new coronavirus, a lot of different things. I think I'm going to settle in on SARS coronavirus 2 because I think it's scientifically accurate and it reflects the close relationship that this virus has with SARS coronavirus 1. And more to the point, when I'm going to slip into a little bit of jargon or nickname and refer to it as SARS-1 and SARS-2. So if I'm saying SARS-2, I'm talking about the new coronavirus, the big coronavirus that we're dealing with right now. And the second thing is, this is a public lecture, so there's a few of my colleagues online, and they can stay if they want. I don't know that they're going to learn that much. Every time I say something like, antigen or epitope or something like that, I get a little bit of an electric shock. So I'm going to try to stay away from the terminology. There's a few things in there in my slides that will I'll have to slow down and explain a bit, but I'll do my best to keep it both simple and accurate. And there is sort of a big picture message here, and the big picture message is on the scientific front. And I mean only on the scientific front, but on the scientific front, things look pretty good. This does, to be very, very clear, this does not mean it's time to end the social distancing at all. And just to remind you where we are, this is month of April, there's going to be about 50,000 American deaths. And with all this social distancing, we're still having 25,000 cases a day in the US. And so we definitely need all the social distancing that we are doing. Now, the basis of my optimism is not that we can go out and have a party right now. Rather, that to a large extent, it's my belief that scientists and science has this in hand. This is a relatively simple set of scientific problems that we can, and we're a long way to completing our work. And what becomes the bigger and more important work is testing, but even more important than that, manufacture and distribution of the vaccines and therapeutics that will be almost surely coming out quickly. So that's the basis of my hope. So why would I say such a thing? Well, the way I see the world is this. HIV is a genius, an absolute genius. And if HIV is a genius, influenza and maybe Ebola virus, these are honor students. But this current virus, SARS coronavirus 2, COVID-19 virus, this virus absolutely flunked Immunology 101. And because of that, we have all this knowledge and we have all the scientific tools we need to defeat it. And again, our bottlenecks are just testing, manufacture, and distribution. Those are important bottlenecks, but scientifically, I think we have this in hand. So why is this virus so stupid compared to, say, HIV? Well, now, if you think about HIV, HIV lives in your body essentially all your life. It has had to master evading your immune system. It knows your immune system better than any human being does. Flu, a very rapidly mutating, rapidly changing virus that can adapt to your immune, to any immune pressure that it encounters. But SARS is different, and SARS coronavirus 2 is different. It's chosen a strategy of saying, essentially of moving from host to host before what we call your adaptive immune response, your high-powered, really good immune response kicks in. And that's your for all intents and purposes for this discussion, that is your antibody response. So it's chosen to move quickly from person to person rather than be very resistant to your own immune system. So that means that it exposes rather than hides the key things, the most important things that your immune system recognizes. It keeps them out there so it can latch on quickly and move very quickly, again, rather than tuck them away and hide them in such a way that makes it difficult to make a vaccine. So this virus is very vulnerable to conventional vaccines and what we call passive immunization. That's the administration of antibodies that we make in a laboratory. So that's one reason SARS coronavirus 2, SARS-2, is so stupid. The second reason comes down to the size of its genome. This virus has a really big genome. Genome here, I mean, is the genetic material that encodes for its ability to replicate. And there's the big word polymerase. I'm going to get to it in a second. But the basic idea here is because the genome is so large, if it's not very accurate in replicating itself, what happens is it introduces, the fancy word is catastrophic mutations. It mutates too many things in its genome so that the next version of that will not replicate. So as a consequence, the way it replicates itself, the way it makes a copy of its own RNA genome is to use what we call a high fidelity polymerase. That's the fancy word. A polymerase is just something that makes a copy of RNA into another copy of RNA. And so this particular high fidelity polymerase has proofreading function, which is to say that it will accurately reproduce itself, and therefore it evolves very slowly. And what this means is that a vaccine or an antibody cocktail that works now is likely to work the next year and the next year. We already have a reasonable sense of that from the original SARS-1 epidemic. We know that an antibody that would have worked at the beginning of that epidemic will work at the end of that epidemic. So there's a few other reasons why this is a relatively easy virus to address, but those are the key ones. So now let's look at the tools that we have at our disposal. Okay, so we have, I think everybody is aware, we have drugs, there's, remdesivir and chloroquine. There's a number of other drugs. Biologics, you may not be as familiar with the term, but this would be proteins that you would inject. So on Jamie's list, we looked at the word Humira. That's an antibody that people with rheumatoid arthritis inject into themselves. These are very powerful, and there's other kinds of engineered biologics that are somewhat antibody-like that I'm going to talk about in a second. We also have, of course, vaccines, and there's 80 now vaccines being propelled forward, and they vary in a huge number of different ways. And then finally, we have convalescent sera, meaning that an individual who got sick, who is now better, their blood, their sera can be infused into somebody else who's quite sick, and that will work therapeutically. But that gives us a lot to talk about in this short talk, and I hope to leave some time for questions, which I think can be most interesting. Now, the good news is that I'm helped a lot because Arnab Chaudhary is going to be talking about drugs in the next talk, and he's both going to be talking about this amazing library that Scripps has, this reframe library of 14,000 compounds that have already been shown to be safe in humans. And I'm sure he's going to be talking about the full array of drugs that are available, and people are discussing whether it's the Gilead drug remdesivir or chloroquine or any other hot topics there. So that makes my life kind of simple, because all of the rest of the things that are on this list have a common element, and that common element is the antibody. And I'm showing you here a very simple version of an antibody. You can see that it's kind of got two chains. It's a dimer of a dimer. So it's got this heavy chain and this light chain. And in these blue colors, I'm representing the place where it would bind the virus or would bind any pathogen. And so all of these other agents that I'd like to talk about all come down to the activity of antibodies. So that leads us, excuse me, let me first, before I move on to a slightly more sophisticated model of an antibody. And in that model, this is a ribbon diagram that roughly places the ribbon where the atomic coordinates are, where the actual atoms of this molecule are placed. And then we superimpose this kind of easy to understand ribbon on those locations. So there is a good deal of reality to this model, and we use these kinds of models a lot of the time. But in any case, we can see that, first of all, the two chains of the molecule represented in this sort of pinkish color and the cyan color. The cyan color is a heavy chain, we call it, and the pinkish color is a light chain. But that's not the important distinction. The important distinction is up here, the business end where the antibody binds the pathogen, in this case, the virus. And then there's a second business end down here, which is where the antibody engages with the rest of the immune system. So both of those components are important. And the properties of this thing down here actually become quite important as we think about what makes a good vaccine or what makes a good passively infused antibody. So that's that's our sort of our first pass at antibodies. And then knowing that antibodies are the sort of important things here. Oh, excuse me, before I go forward, I want to point out that that Scripps, when it built this beautiful campus in Jupiter, Florida, was wise enough to realize that antibodies were very important. So wise were they that they actually built a statue to the antibody as you come into our entrance. So anytime you're visiting our campus in Jupiter, Florida, you can take a look at this antibody and realize we knew these things were important, and they are indeed important. I would even say they're the hero of our story here. Sort of moving back to antibodies, the three key questions that I want to look at today are, how do we make antibodies? Okay. And then how do antibodies stop the virus? And then how will all of this help us end the pandemic? And so let me try to get to each of these questions. And let's start with, how do we make antibodies? And this, I was thinking that one should, and we could give a semester-long class on the process of generating antibodies in the human body. But we can actually reduce it to very simple principles. And the simple principles are the same principles that we know evolution works with all the time. Basically, you diversify, you make a lot of variants of something, closely related variants of something, and then you impose some selection onto that. And then you take that which you have selected and you multiply and diversify again. And then you select again. It's very, very straightforward. And what our immune system has done is they've taken these wonderful principles of evolution and they've internalized them into your body. So we're doing that exact same process in your body. That is the process by which species evolution also occurs. And there's a little subtle difference here. There's a lot of differences. For example, antibodies don't have sexual selection, so they don't, in general, although you can somewhat argue they might, two different antibody producing cells don't exchange material and come up with a completely different antibody. But the most important distinction is evolution writ large does not, is kind of agnostic on the principles by which it is selected. That is to say, it might be that the mold is holding tighter onto your bathtub. That's the principle by which selection can occur, or having better plumage, or you name it. Evolution in general is agnostic, but the immune system is not agnostic. What the immune system is selecting on is the ability to bind more and more tightly to something, a pathogen, a virus, that appears to the immune system to be dangerous. And that word dangerous is actually quite important because many people view the immune system as something that distinguishes self from non-self, and that's true. But the immune system also needs clues that a particular thing is going to be dangerous. And there's a lot of clues. And those clues are things that people who manufacture vaccines also try to emulate. So we not only try to provide something that looks like the pathogen, but we try to emulate those danger signals as well. But in any case, what we do in our bodies when we make antibodies is we make a lot of different variants. We select them for their ability, or they are selected for their ability to bind a pathogen. Those get again amplified and diversified and selected again, on and on. And that's the process by which you get what we call high affinity, tight binding antigens, antibodies to bind what we call an antigen, which is just a piece of a virus that the antibody binds. Okay, so that's how we make antibodies. One semester course down into one slide. The next question, which is a little bit more interesting still, is how does an antibody stop the virus? And actually, it's not more interesting, it's kind of simple. And the first thing to do is to start with this model we've both grown to hate of this virus. But the thing about this model, we're really sick of this model, is that it's actually scientifically quite good, because it shows exactly what is really truly exposed on this virus. So you can see in the gray here, if I can point to it, that is your lipid membrane. And then the little orange and yellow BB things, those are representing proteins that are not very well exposed. They're not that important. For those of you who care, this is the M protein, and that is the E protein. Yellow is the E protein, orange is the M protein. But what the real important thing we need to focus on here is the red protein. And this particular model of the red protein, if you look carefully, is good. It appears to have been based on an actual cryo-EM structure. And what you can see is that it looks like a triangle, and it's a triangle because it's a trimer. There's actually 3 different identical versions of the same protein coming together there. And then in the middle of that is the place, right dead center in the middle of that is the place that that protein will bind the receptor, as I'll show you in a second. But let's get rid of that horrific model, because gosh, we see it way too many times, and just sort of simplify it down a little bit. Again, so we just have these red spikes. I've sort of gotten even rid of the trimer part of that, and just the gray lipid membrane. Nothing else is important, okay? And we're gonna, next few slides are just gonna walk down in a very cartoony way. By the way, the cartoons were modified from some nice graphics from the New York Times, so full credit there. A cartoony way, exactly how the virus gets into the cell, and therefore, and then how an antibody blocks that process, giving away the punchline here. But before we do, let's remind you that there's an inside of the virus as well. And so again, you can see the red spikes, the most important part, these yellow and orange proteins, not so important for our purpose. What remains important is the RNA in the middle. That's important in part because a good deal of our testing is associated with recognition of that RNA. And then what the New York Times failed to show you is a protein associated with that RNA. It's called nucleocapsid. And that's protein's only worth mentioning because that is a useless, let me say it this way. Your body tends to make a lot of antibodies to that protein. These are useless antibodies, but they're an important component of the serological testing that's going on. So the internal protein called nucleocapsid associated with RNA is a bad antigen. but it is being used to detect whether people are seropositive, meaning whether they've seen the virus or not, whether their immune system has seen the virus or not. So, so that's the virus as we can see it. Now we're going to get to the question of how do antibodies stop the virus? And the first thing to tell you is that this virus, through its spike proteins, through these red proteins, bind this little, looks like a wrench to me, but it's a molecule that I'm kind of proud to say that my wife, Hedy Shea, and I identified back in the day for SARS-1. It happens to be the same receptor for SARS-2. And this, what I'm showing you here, is the lipid membrane of a cell. And it's, in this case, because it's expressing ACE2, it might be a cell in your nose, or it might be a cell in your lungs, or it might be a cell in your gut. But it's only on a subset of all your cells, but it is on important cells for the virus to replicate. And you can see that the first thing that happens for the virus to get into the cell is it kind of latches on to this receptor. We call it ACE2. So let's pause for a second and ask ourselves, well, what is ACE2? So we know a lot about this molecule. The most important thing to say is, in this context, is that it's necessary for both SARS-1, the original 2002-2003 SARS, and now SARS-2 to replicate. So without this molecule, the virus will not enter the cell and it will not replicate. It will be stopped cold. The other thing about angiotensin II is its normal day job is to regulate your blood pressure. And so many of you have heard of ACE inhibitors. These are actually targeting a molecule called ACE1, not ACE2. So ACE1 is something that cleaves a little peptide called angiotensin. The details aren't so important, except for the fact that when ACE1 cleaves angiotensin, it signals the body to constrict its blood vessels and raise your blood pressure. So if you inhibit ACE1, your blood pressure goes down. Now, it turns out ACE2 actually counteracts the activity of ACE1, and this makes it very complicated as we think about the role of high blood pressure in COVID pathology. But in any case, that is the day job of ACE2, is to be part of the system for regulating your blood pressure. Now, where is ACE2 expressed? Well, it's expressed very importantly on your, what we call your upper respiratory tract, in your nasal epithelium, in the nose lining in the cells lining your nose that allow you essentially to smell. And they communicate with neurons that allow you to smell. They're also very importantly in your lower respiratory tract, in something we call type 2 pneumocytes. These are the cells that help to provide mucus in the region of your lungs in which gas is exchanged. And that's also critically important. And in addition to that, it is in your gut, all over your GI tract. And that we know because most coronaviruses actually transmit through the oral fecal route. Almost for certain, we got this virus because a bat pooped on either a human or an intermediate species that then transmitted to humans. Oral fecal is the primary route by which these classic coronaviruses transmit in the wild. It's also importantly on your heart and your intestines, and we can see that from the pathologies of the more serious COVID cases, because there's a good deal of heart pathology and also intestinal pathology that is associated with this, and that simply maps to the location of ACE2 on these cells, meaning that there is infection and inflammation in those tissues. I want to get back for a second to talking about the nasal epithelium and then the lower respiratory tract. We always understood that ACE2 was on the lower respiratory tract, but it was only in this new infection we began to appreciate the degree to which ACE2 is present in your nasal epithelium. And this is very important for a couple of reasons. One is it helps to understand the very unusual distribution of disease severity that associates with COVID-19. And which is to say, as you all know, I'm sure, that some people get very sick from this virus, and a lot of people don't get very sick from this virus. They have what we call upper respiratory tract infections. You got a nose and a little nasal drip and maybe a cough. Whereas if you get into your lower respiratory tract, you could have inflammation and very serious pneumonia, and that's the beginning of how you might die from this. So the idea that's in your nasal epithelium is very, very important, because that talks about how easily it can be transmitted. So just to take an aside, flus that only attack your upper respiratory tract, they transmit efficiently, they don't cause serious disease. Flus that attack your lower respiratory tract don't transmit efficiently, but they do cause very serious disease. Flus that do both cause, transmit and cause serious disease. And SARS-2 is like those flus, only worse because we've never seen it. There are some other interesting points about ACE2. ACE2 tends to express higher when your lungs get insulted. If you've been exposed to a lot of pollutants or something of that nature, or you smoke, you're going to express more ACE2. It's a little subtle in smokers because the type 2 pneumocytes express about the same or a little less ACE2, but it starts distributing to your type 1 pneumocytes as well as you smoke. So good time to start thinking about not smoking so much. Super interesting, just out from the Broad Institute, children, as it turns out, young adults and children, do not have much ACE2 in their lower respiratory tract. They do have ACE2 in their nasal epithelium. This makes it very clear why they do not get as sick as many adults do. And it also makes clear that at least young adults, meaning teens, are still nonetheless going to be able to transmit this virus quite efficiently. So that's worthy of note, and it's an issue as we're talking about opening schools, etc. There's something in here for the scientists, just for a second. There's a molecule called, there's a set of molecules called the type 1 interferons. This is part of your antiviral response. And normally that's a good thing. It helps control a virus. Unfortunately, ACE2 is also in expression, is induced by these type 1 interferons. And this creates a potential feedback loop that we see with other viruses as well. Typically what happens is the infection creates a mechanism by which infection becomes more efficient. And the nastiest viruses have that property. HIV has that property, as do a number of hemorrhagic fever viruses. A final point here on ACE2, and then we'll wrap up with ACE2, is ACE2 utilization can tell you whether an animal species is infectable by the virus, and gives you a very good hint on whether that animal species will be a likely source of the virus, that it was an original reservoir, or whether, for example, it might be a reservoir later. So for example, if you find out pigs are a reservoir, you may want to do a little bit more carefully, because you know that their ACE2, the ACE2 version in pigs is efficient for SARS coronavirus 2. You might want to do some more monitoring pigs, and we do indeed, and the pig receptor appears to be reasonably efficient. So you can learn a lot from a receptor, I guess, is the main point here. And then, but sort of circling back to what the question is, how does an antibody stop this? Well, let's talk a little bit more about the interaction between the virus and the receptor. So again, we've got this red spike interacting with these greenish wrenches that I'm labeling here ACE2. And just to throw a little bit more science into this, it looks like a very simple interaction, but it's not. It's a very interesting and delicate dance between the red entry protein and the wrench-like receptor. And indeed, that red entry protein is capable of a lot of activities, and most importantly, it undergoes a shape change. And why does it undergo a shape change? That shape change provides energy for an otherwise energetically unfavorable event. And that energetically unfavorable event is the merging of the viral membrane with the cell membrane. They're both negatively charged. They don't want to be together. So you need to get energy to overcome that barrier. So you need that red spike protein, and you need a signal in the form of that wrench-like ACE2. And so ultimately, the events that follow is the membranes of the virus and the cell merge, and that gives the viral genetic material access to the cell, or we would say the cell cytoplasm, where the RNA of the virus is delivered, and that immediately initiates two events. It initiates one event, which is the formation of new viral proteins, and it initiates the formation or generation of new viral RNAs, and then those go on to assemble to make a whole new virus. So you stop it at the beginning, and you can stop the whole replication process. There's one other important point. With these viral replication, different drugs and different inhibitors stop at different places. So many of you have heard of the Gilead product, remdesivir, and that stops the replication of the viral RNA. That's very important. Stopping the entry process is especially useful because compounds that do that never have to enter the cells. And so they are typically safer than compounds that have to get into your cells. And so that's important if you want to start thinking about inhibitors being used prophylactically. And that's the basis of what I'm going to get to later, the point that antibodies will be very, very useful in protecting people from infection, as well as treating an ongoing infection. Bottom line point here is the spike. The red thing is a very delicate machine. An antibody can get at it, and it can be easily blocked. Okay, so now a little bit more subtlety on the issue of antibodies. So we already discussed if your immune system sees a pathogen or a vaccine, it's going to start making the antibodies by that selection and diversification method I mentioned. But then we got to start thinking about what these antibodies do. Now, the most important antibodies bind the most important or the most accessible protein on the virus, and that's the spike protein. And when it does so, and it blocks the activity of the spike protein, we call that neutralization. And these are the best class of antibodies, I'll say in a moment. So the blue part of these antibodies are the things that bind the spike and other parts of the immune system, other parts of the virus. But there's another part of the antibody that I mentioned. These are, fancy word, effector function, things that recruit the effector function, and these effector functions recruit cells to clear the virus or to kill infected cells. So you can actually have an antibody that is not neutralizing. I mean, it doesn't really stop the virus from doing anything, but it's still helpful to you. And so we can think about antibodies actually in three classes now. We can think of antibodies, in this case, that directly block the interaction between the spike and ACE2. These are neutralizing antibodies. These are really the best ones. But we can also think of antibodies that bind other places on the spike, and these could be useful because they can signal other immune cells to come and maybe eat up the virus, or if they see an infected cell because it's poking out a lot of virus, to go ahead and kill that cell and stop that replication process. But there's also a third class of antibodies, and those are the antibodies that are essentially useless and sometimes even harmful. So we want to keep an eye out for those. And unfortunately, the virus wants to guide you in the direction of making those useless antibodies, and it does, perhaps by making particular regions, antibodies against particular region of the S protein, and certainly by making antibodies against the nucleocapsid, which these antibodies are useless because the nucleocapsid is inside the virus. The antibodies can't get there. And the only time the body sees the nucleocapsid is when it's viral debris. So just thinking about those three classes of antibodies will help you think about what can make a good or somewhat good or not very good vaccine, and what a good antibody response is in the first place. So now I'm just sort of going to move on from that sort of background to talk about these three classes of things, all of which depend on these antibodies. So let's start with something we've been working on quite closely right now, and that is vaccines. And let me sort of tell you what it is we think we're doing with vaccines. we would say at Scripps, or certainly at, on this campus of Scripps, we're not necessarily making a vaccine. What we are doing is telling people who make vaccines what is the best thing to target and how best to target that thing. So that's what I'm going to tell you about next. That's some of our work. And we, as a consequence of that work, we have an opinion on that. So let me start again from this up here in the corner here, this horrific but scientifically accurate model of the virus, and move you over to an actual real electron microscope image of the virus. You can still see these spikes, these what are called corona for crown, surrounding these viruses. And then let's sort of zoom in a little bit more and imagine that this is those same viruses. That's the lipid membrane of the virus, and these are those same spikes. And this is an actual ribbon diagram of a molecularly accurate form of the spike protein, the SARS coronavirus 2 spike protein. Several groups have made this. I'm using a group from a group at the University of Washington right now. So let's even zoom further on down below here and notice that there's actually two forms of spike that people have been able to observe with this cryo-electron microscopy. One is this sort of closed conformation. And the way you know these closed conformation is this red region and this orange region and this yellow region. They're all in the same orientation. Now these are actually the same piece of the protein from different, what we would say, monomers. So this, as I mentioned, is a trimer, and these are each a component of one of what we would say protomers or monomers of this trimer. So I've, in this case, labeled one is red, one is orange, one is yellow. They're all the same, except for the idea that in some structures, we also see that these things pop out. And they pop out because they are getting ready to engage the receptor. So this is actually pretty floppy in here, surprisingly floppy. This is something that somebody in the HIV field would never see, or similarly flu or Ebola. You would never see this kind of thing, kind of like just kind of loosely hanging out and showing you, its most vulnerable location. But that is what the virus is showing you. Again, it does so because it doesn't really care about your immune system. It's going to move on before your immune system really kicks in. What it really cares about is efficiently getting to the next host. And then this is a picture of this little region here. We all call this the receptor binding domain, or the RBD. And it's an RBD because, of course, it binds the receptor. And here is indeed a picture of the receptor I'm showing in gray. This is ACE2, the one I've been showing you as wrenches before. This is a more molecularly accurate ribbon diagram of this molecule. And you can see the relationship between this receptor binding domain, which is the same as this guy down here, which is the same as this guy down there. And you can actually pick it out on that horrific model of the virus everybody's showing. But here it is. And so one of the things that we wanted to ask was the question, to what degree is that receptor binding domain sufficient as a vaccine? This is a very important question. And the reason it's important is because it's a heck of a lot easier to make this small piece than the large piece. So let me just show you. The large piece here is 20 times bigger as a trimer than this small piece. That means we can make 1/20th the versions of this big thing than we make of the small thing. So whatever vaccine you're using as the delivery system, you're going to make a lot more of the small thing than you can make of the big thing. And also, it's cheaper and easier and more scalable to manufacture. So if you're thinking about 7 billion vaccines or 8 billion vaccines or 4 billion vaccines, this becomes much more tractable than this whole thing here. Now, we're going to go into why most of the big groups have started in on using the whole big thing here, but we are making the argument, and I think we have data supporting that, that suggests that the small thing is sufficient. So here's just a piece of the data. For the scientists who shouldn't be on this talk in the first place, you can go down to bioRxiv and look this up, or hopefully that'll be published soon, and we'll be able to show you that, or email me, I'll send you a copy. But the bottom line point here is the immune response that we get from this small thing are way more than enough than what we would expect to protect animals and humans from infection. Now, how do we know that? Because what we did is we took the sera, the blood, from these small animals, they happen to be rats, and we compared that to blood from the same rats that have never seen the vaccine that we're giving them, the vaccine that we're giving them. And we doped in a certain amount of a very potent inhibitor that I'm going to tell you about in a bit. And that potent inhibitor is just a form of the receptor, a dimerized form of the receptor, some fancy tricks, but nonetheless a very potent inhibitor. And we know from our work from HIV that if we have an individual or an animal that makes 100 microgram per mil of something analogous, that animal is superbly protected from the virus. And so the immune responses that we're able to raise here live somewhere between that 100 microgram per mil level, sorry about the numbers and the units here, but just somewhere between a lot and a whole lot of a very good inhibitor. So the bottom line point here is this piece of the spike protein, that piece of the red thing you've been staring at all along, is sufficient as a vaccine, and it's a lot easier to make. And for your aficionados out there, we were able to show that this does not have one of the risks that might be associated with a vaccine that combines neutralizing and non-neutralizing antibodies. So remember, this is the primary neutralizing epitope. And so this particular vaccine, with some great work coming out of Hetty and Che's lab, does not carry the risk that we don't know yet, but we worry is associated with the larger vaccines. So that's my agenda, and the thing that we think is true, and I think that our strongest case is the ease with which this particular thing can be produced if you're thinking about making this in scale, as we are. So that said, a lot of people are pursuing the alternative route, and I want to be a little bit careful and fair to the alternative perspective here. So even though I'm saying that the RBD is a great vaccine because it's showing you one very important region to target, a neutralizing epitope, a part of the region that if an antibody binds, it blocks the function of the spike protein. There are a few other points to make. There are other parts of this molecule that if you bind this trimer, this spike protein, that if you bind, you can block. So there are other neutralizing epitopes that we're not using. And in addition to that, there are places the antibody can bind that can elicit those other, what I called effector functions before, that can recruit the immune cells to clear the virus. So some of those will not be made and elicited. We will not get those in humans vaccinated with just this receptor binding domain. And indeed, those effector functions are much better. They work much better if you have many antibodies hanging off a target, rather than, as you would expect with ours, meaning that RBD vaccines, we're not the only people doing it, you would only get antibodies to this region here, the most critical region, but it is very, very possible that you may want other antibodies as well. So the concern, though, is that the early vaccines, now based on these soluble trimers, will not provide enough of an immune response because they do not express sufficiently. We know, for example, Moderna's vaccine is an mRNA vaccine, and that is very sensitive to how much protein gets made. And it takes a lot of the cell's energy to make this big, complicated protein over there, and not very much energy to make that smaller. I'm going to tell you how many amino acids are in that. It's less than 200, whereas the big thing has 3,600. Another way to think about this, though, is in terms of a prime and a boost. That is, you give one of these agents that allows you to focus on the most important region of the molecule, and then you give the other agent, and then you can sort of get the best of both worlds. And you can certainly rebalance the immune response so that it is more neutralizing because it's presented more of this receptor binding domain. Anyway, those are the issues. And of course, we're running this gigantic test in vaccinology and immunology right now. And it's going to be very, very interesting to see. Again, though, scale of production, ease of production, cost of production, rapidity of production, these all lean us in the direction of the receptor binding domain. Okay, let's now move on to something else, something that Scripps, both in California and Florida, do very well, and that is biologics. And I got to give a shout out to our colleagues in California who are literally the world leaders in the development of antibodies against HIV. And that's led by Dennis Burton and some amazing faculty on both campuses that are helping him develop antibodies against HIV, against malaria, against Ebola, et cetera, and now against SARS-2. And I genuinely think these antibodies are very, very important. There is another antibody-like molecule that I think is worth mentioning as well. I already mentioned it, which is an antibody-like form of the receptor ACE2. And this has some advantages as well that we're going to go into. But the basic bottom line, and I just want to stop here and pause and get everybody to understand that a cocktail of antibodies against this virus can be extremely useful, not just for treating individuals, but for protecting people, because we know, the world knows, scientists know, how to engineer these antibodies, one, to be safe, but also two, to be long-lived. So we can take that one of those business ends of the molecule, make a little tweak to it, and have that antibody live in your body for months. That means with a single injection of, say, 2 antibodies or 3 antibodies, an individual, say a healthcare worker, can be protected the virus before we can offer them a vaccine. And I think this is super important, and I hope and I know that there's a huge amount of work going on around the world to develop the best of these antibodies. One of them I'm very sure will target the receptor binding domain, and that may want to be complemented by an antibody against other regions, or 1 or 2 antibodies against other regions, and or this molecule I'm going to talk about, ACE2IG. But I would say that the possibility of recombinant antibodies helping in this current situation, helping rapidly in this current situation, is very high and the source of a good deal of my optimism at the moment, scientific optimism. So there again is our model of the antibody. And just to remind you, again, it's got 2 business ends, where it binds, in this case, the virus, and where it associates with the immune cells. But also I would suggest to you that, or I would remind you that this region down here is very, very useful for one other purpose. It extends the half-life of these antibodies in the body. So an antibody, a regular protein, lives for hours or maybe a day in your body, but antibodies live on average for about 21 days in your body, half-lives. We can change this so it now lives for 2 to 3 months in your body. And that's super important. And I have no doubt that the first antibodies that are rolled out for healthcare workers, for people who are vulnerable to this virus, will have that change. Now we can think about one other antibody-like molecule at the same time. And that is something where we take the virus binding piece of the receptor and fuse it to this business end of the molecule. We call this ACE2IG. It's something that we showed works very well in the SARS-1 case back in 2002, 2003, 2004. It's really quite a simple molecule. There are some nooks and crannies, and we're working very, very hard right now to increase its potency and also to increase its half-life, because actually, even with this little guy here, and even with a little tweak that allows it to live a long time, it lives not as long as many of the best antibodies here. So what would be ideal is that this thing lasts almost as long as this thing here. Then there's certain advantages that I'm going to tell you about in just a second for this molecule relative to this molecule. But which one ever, doesn't matter which one of these is most effective. Either of them would be very, very useful at treating infected people, and very, very useful for protecting people who are on the front lines, who might be especially vulnerable to this virus. So yeah, that's super important. This is just data showing you from the old SARS-1 days that this is a very, very potent little guy. So sorry about the units, these are nanomolars. It doesn't matter. It means that it is potent. And this is with a model virus. The scientists will know that this is a retroviral pseudovirus, and then this is infectious virus. And in the model virus, you get something on the order of 2 nanomolars, and the infectious virus, you get something on the order of 10 nanomolar IC50. So that, in our world, is very effective. So the question is, well, which of these is actually going to be better? Well, to be honest, I'm not sure it matters that much, because what matters is just us getting these things made quickly. But I want to go through that just for a second here and tell you that this ACE2IG has some advantages over antibodies because it works against, and it will work against, all SARS and SARS-like viruses. So almost for sure, any good antibody that works against SARS-2 will not work against SARS-1, and it will not work against any future SARS-3. And there is the worry that a single monoclonal antibody will not even work against SARS-2.1, meaning the virus has acquired a mutation, one of those rare mutations that it might acquire over the next year or two. So there is that worry. So that, in contrast, the virus always has to bind ACE2, and the degree to which it binds ACE2 efficiently is also the degree to which it is pathogenic. And as I showed you, it's very potent, as we've shown with SARS-1, and we can increase that potency with a lot of tricks. And most importantly, you only need one. So I think most people would suggest that if you're going to think about monoclonal antibodies, you're going to need some sort of cocktail, maybe 2, maybe 3, just to be safe, that there isn't some sort of breakthrough, the virus doesn't change in such a way as to knock off that antibody. However, as I mentioned, you've got a shorter half-life right now, and so that half-life needs to be improved before we can really think about it as useful for prophylaxis. I would suggest to you that both the antibodies and this thing should be quite useful for therapy, and that we should be making them as quickly as possible, and therein lies the rub. And the most, I'd sort of be remiss if I didn't mention this final tool at our disposal. Now, this tool has some serious problems, but it has one very important advantage. And the very important advantage is we can do it now. What this is the ability to take from an infected individual their blood, their serum, their antibodies, and infuse that into somebody probably in the ICU and save their lives. And we know this works. We know this works because there's been some amazing doctors in Boca Hospital, and I'm getting messages, I'm running way over time. So in Boca Hospital who are using this effectively, and then the only key question is, what makes good sera, what makes bad sera, what makes intermediate sera? So just to wrap up here, where are we? So convalescent sera, this is an immediate high priority that will save lives in the short term as a stopgap. We need to be smarter about making it more effective, knowing what is more effective. Biologics, this is a straightforward path. There's real hope. We should be using cocktails of 2 or 3 antibodies, make them quickly, and possibly comparing them with ACE2IG. Vaccines, easier than for most viruses. Most approaches will work. A vaccine will ultimately replace an antibiotic will ultimately replace these antibody cocktails, but the first vaccines may not be sufficiently effective for those who might need them the most. But ultimately, the science is nearly done, basis for the hope, and that it really comes down to testing, relatively short-term prospect, manufacture, and distribution. And if there's time for questions, I can take questions.
Thanks a lot, Mike. This is Jamie. We have a little bit of time for questions.
Yeah.
I think I'm going to have to aggregate the questions. We've got about 100 questions that people have typed in, and maybe I could ask one that kind of groups together a bunch of questions. I think people are pretty interested to know, so now you have this great RBD antigen. What's the timeline that would be for you to turn this into a vaccine and then have that vaccine deployed into the general populace?
Okay, so that's a terrific question that I can't give a straight answer to. So there are two pathways we're taking. Pathway one is to publish our result and make sure those individuals with the kind of GMP resources listen to us and take our advice. The second thing is we're trying and we're enlisting individuals to take one form of manufacture of this class of vaccines and move it forward. There are certain advantages to the approach we're taking, and that is we can shave off about 4 months of the otherwise year to year and a half timeline for the development of these things. So that's a key advantage. But if push comes to shove, we're certainly out 8 months, but maybe more optimistic than a year and a half.
Right. Thanks, Mike. Now, here's another question. You spent a lot of time talking about ACE2, and people are generally aware that ACE inhibitors are used to treat hypertension. And so there were a lot of questions about the interaction of these kinds of medications with the vaccine and with the virus. And maybe you could say a little bit.
I would say that's an enormously complicated question because there are many ways in which both the use of those inhibitors and their absence can contribute or alleviate the COVID severity. So I'm following, knowing that this is an enormously complicated system and that there are pieces of the system that we do not fully understand. I would absolutely follow the advice of medical doctors that I respect and follow, who suggest that at this moment, the best process is just stay on your blood pressure medicine, because we know that high blood pressure is a risk factor, and we don't know that any of these drugs are.
Okay. So let's see, find another question. So another question was about the mutation rate. So you talked about how there's a natural mutation of the virus. And if you develop a vaccine, does that affect the timing the time over which that vaccine might be effective?
The most important determinant of the timing in which a vaccine will be effective is its inherent durability, which is a problem for many classes of vaccines and will likely be a problem for this vaccine. The tendency of this particular virus to mutate is less of a worry than the inherent durability of a vaccine, for the simple idea that you make many kinds of antibodies. When you make a vaccine, you make many different antibodies that bind in slightly different ways. And so even if you acquire those one or two mutations that would knock off a single antibody, you still have other antibodies that continue to bind and prevent the infection. So a vaccine tends to be broader than any single antibody. And therefore, the mutation of the virus is less of a concern of the vaccine than it would be with a single monoclonal antibody. But we have this at the back of our minds, the inherent durability of vaccines, which is an issue and may require boosts in a year or two after the initial vaccination.
Okay. Another general theme question would be, people are interested in the relationship between viral loads. So you might get one virus, you might get a lot of virus, and how does that impact your immune response?
The first answer is dose matters. Dose absolutely matters. I think anybody who's been following the news can understand why apparently young and healthy healthcare workers are dying of this thing, because they were initially exposed to a large dose. I kind of say you want to flatten the curve in your own body. It matters in part because it matters which tissues get infected and how much of those tissues get infected before your immune system can kick in to help control them. And so you've got, just to give some examples, you've got 5 lung lobes. If only one of those lobes is infected with one infection and maybe one virus that's taken a while to get off to its logarithmic phase of growth, you're in a better shape than if several of your lung lobes are infected and doing the same thing. So at all points, try to minimize your dose of exposure as you're protecting yourself from this virus. So I think that, I hope that answered the question.
Okay, yeah, thanks, Mike. And maybe another theme question is there's a lot of evidence that there are benign coronaviruses around that can be taken for a common cold. And
yes.
And so how are we going to sort out the infections from the novel coronavirus, COVID-19, and these other benign coronaviruses? And how are we going to track people's immunity when we start doing antibody testing and serological tests?
Right. So the concern is that there are false positives, and the current tests have a good deal of false positives that are distorting some of the data that some people are using to suggest that more people are infected than probably are. So these tests have high false positives, and the thought, but not certainty, is this is due to exposure of other coronaviruses that are related. And then it sort of gets into the weeds. So the nucleocapsid is more conserved among coronaviruses, and the easiest and most sensitive tests right now are recognizing the nucleocapsid, but they also have the lowest selectivity, meaning that they're higher false positives. Serotests that rely on the spike are more discriminating, but not everybody makes good antibody responses against a spike. So I think I'm not giving a complete answer to this, except to say that there's a good deal of sorting out to do. And it may be that a test that combines recognition of nucleocapsid and spike together would give you the best combination of low false positives and low false negatives. And so that is indeed a problem. Now for vaccination, it's not a problem, and it might even help a little bit in the margins. But in point of fact, the S protein is quite different among the mild coronaviruses and the SARS-like coronaviruses.
Okay, Mike, thanks very much for this really engaging lecture. I'm getting lots of kudos and positive comments in the chat for your talk. I think this is just a challenging time, and we are all waiting for the solution to this problem. We're all trying to understand the tools that are available, the therapies that will become available for us to combat this. Most of us are all at home at the moment, and we're itching to get back to our regular lives. And it's through the efforts of you and other scientists at Scripps, as well as around the country and the world, that we will be able to make that transition. So I want to thank everyone for hanging in there. There will be, there's lots of clapping going on. There will be, the slides will be posted on the internet, and I'm sure that Dr. Farzan would be happy to answer questions. We really appreciate you tuning in, and we hope you will join us for the next front row lectures. Stay tuned for more information about that. So thank you all for coming, and have a great day. Goodbye.